Welcome everyone to the Genetic Engineering and Biotechnology News webinar series. Our presentation today is entitled, Peeking Under the Hood of Cellular Signaling Machinery with Targeted Proteomics Assays. I'm Jeff Bogaliskas, Technical Editor for GEN, and I'll be the moderator for today's webinar presentation. Proteomic profiling methods are aimed at quantitatively measuring the differences in protein expression across samples. Results from profiling experiments can often serve as a foundation for the future study of quantitative changes or modifications in target proteins. When profiling results are combined with genomic analysis data, researchers can observe a more comprehensive view of the dynamic changes taking place within cellular systems. However, limitations in proteomic methodologies have plagued researchers and hindered them from taking advantage of a vast array of tools available to their genomic colleagues. To address these issues, scientists have begun to develop targeted proteomic assay approaches with customized automated workflows to increase speed and reproducibility. Let's meet our panel so they can discuss the experiences with proteomic assay development and automated sample preparation platforms. Our first speaker today is Dr. Jason Russell, scientist at Agilent Technologies. Dr. Russell will provide a brief introduction into automated workflows and how Agilent's AssayMAP system can be utilized for a variety of laboratory methods. The second presentation will be given by Dr. Jacob Jaffe, Director of the Link's Proteomic Characterization Center for Signaling and Epigenetics, as well as Associate Director of the Proteomics Platform at the Broad Institute. Dr. Jaffe will discuss his work in targeted proteomics profiling and how automated biochemical workflows are integral to his current research. Before our speakers get started, I want to encourage the audience to submit questions for our Q&A session at the end of the presentations. We will try to answer as many questions as we can, so simply type your question into the Q&A box on the left-hand side of the screen and hit submit. So, without any further ado, let's begin our webinar. Jason, the floor is now yours.
Thank you for the introduction, Jeff. Today it's my pleasure to introduce the listeners to Agilent's approach to automated phosphopeptide enrichment workflows enabled by the AssayMAP Bravo automation system. There are three main technological components to the AssayMAP automation system. The first is our portfolio of microchromatography cartridges. Included in those cartridges are titanium dioxide and a new cartridge that we launched this summer. It's an iron NTA cartridge for phosphopeptide enrichment. The second key technological component of the AssayMAP system is the use of positive displacement pipetting for doing solid phase extraction. So in this regime, liquids are actively pipetted into syringes, and then microchromatography cartridges are mounted, and we dispense liquids through those cartridges. And the third real technological component of AssayMAP is the way that Agilent has approached the use of automation software. The AssayMAP software has an incredibly simple user interface, really designed for analytical chemists as opposed to automation engineers. And this design allows someone like me, who's an analytical chemist, to go in and do method development on an automation system without the requisite the requirement to learn what can be incredibly complicated automation language to do the programming. And along with these new iron NTA cartridges, we've updated our automated phosphopeptide enrichment application, and we've also included in our software an opportunity to customize this cartridge with metals other than iron. This is a closer look at the automation platform, the liquid handling head of the AssayMAP Bravo. And as you can see here, there are 96 syringe probes, and these probes are specially designed to mate to AssayMAP cartridges. And with these syringes, we can do things in addition to solid phase extraction. We can do normal pipetting with pipette tips, or we also can use the bare syringes to aspirate and dispense liquids. And here's just a view of the bottom right-hand corner of the construction of that AssayMAP syringe. The maximum volume of these syringes is 250 microliters. And all 96 syringes are driven in unison with this particular liquid handling head. Probably the critical component for the phosphopeptide enrichment platform is the use of these microchromatography cartridges. And here's a general design of AssayMAP cartridges. It's a two-piece design with integrated bed supports, and each one of these cartridges is slurry packed, and the bed volume is approximately 5 microliters. And to give you some idea of scale, an AssayMAP cartridge is only a little bit larger than a United States quarter. One of the things that Agilent prides itself is the quality of its instruments and its consumables, and that also applies to the AssayMAP cartridge. Every single cartridge is back pressure tested, so we have an expected pressure drop for each of the resin types that are slurry packed into these cartridges. So each cartridge is slurry packed, we get a back pressure trace as part of the quality control process. And an additional level of quality stringency is we have a vision detection system. So each cartridge is passed through this vision detection system, and it can detect the presence of voids, as you can see on the bottom right-hand corner. So that's the cartridge that would automatically be rejected. And this is an additional way that we can guarantee the type of reproducibility both Jake and I are going to demonstrate today. One of the greatest assets to the AssayMAP Bravo platform is the protein sample prep workbench software. And this is the software that we use to navigate and to conduct AssayMAP protocols. And we've constructed this workbench software and separated it into different libraries, workflows, applications, utilities, and literature libraries. Example of some of the workflows that we've currently enabled with the AssayMAP platform. I'd like to highlight the phosphopeptide enrichment workflow. And the way that Agilent approaches these workflows on the AssayMAP Bravo system is we take all of the steps that most people doing a phosphoproteomics enrichment workflow would need to go through digestion and cleanup and the enrichment. Now, not all of these steps are integrated into one smooth transition, but we've provided each of the steps. So, for instance, you could do in-solution digestion, lay out the deck, and then once digestion is complete, you could then go to the phosphopeptide cleanup application, reset the deck, and run that protocol. And so all of the steps are assembled in an easy navigable place. And we've also provided additional tools to help method developers, including the addition of calculators to help reconcile labware volumes, and we've also provided quick start guides, full user guides, in addition to a host of very simple liquid handling utilities to help users transition between each of these steps of the phosphopeptide enrichment workflow. If you don't need to conduct an entire workflow, you can operate applications independent of one another, including the phosphopeptide enrichment updated version 2.0 application. And here's the phosphopeptide enrichment version 2.0 user interface. So the way that we've designed the software is to really have a focus on method development and very task-specific design of our automation protocols. So what we ask users to focus on are typical method development steps, in particular for solid phase extraction in the case of phosphopeptide enrichment. So this would include volumes and flow rates and particular steps that you do or do not want to do. And a lot of these can be enabled or disabled with a simple click of a mouse button. In addition, volumes and flow rates, the R&D group have provided very reasonable starting points with the default values that will work for most applications. So most of the time we can set one of these systems up in a matter of hours and can be running validated methods or creating validated methods in the course of a day. And now I'm going to get into a little bit of the chemistry and how the group of people in AssayMAP R&D, what we've done to characterize the performance of these cartridges. And one hallmark of AssayMAP cartridges is our ability to elute in incredibly small sample volumes, our elution volumes, and that's enabled by our ability to strictly control the flow rate. And here's an example where we took some alpha casein digest. This is a model phosphoprotein. It's heavily phosphorylated. We loaded this phosphoprotein digest onto an iron NTA cartridges and elute it with 1% aqueous ammonia, and we measured the signal of the phosphopeptides. And all of the phosphopeptide signal had eluted in 10 microliters. And amazingly enough, the majority, over 99% of the signal, was only measured in 4 microliters of eluate, which is a little bit less than the volume of an AssayMAP cartridge. We've also looked at AssayMAP-specific types of considerations, including how the sample loading flow rate affects both the number of phosphopeptides that can be identified as well as the selectivity. We can see here a sample loading flow rate from anywhere from 2 to 6.5 microliters a minute really doesn't affect the ability to identify phosphopeptides to a large degree, nor does it affect selectivity. Selectivity across all of these sample loading flow rates was about 90% or better. We've also looked at how both the type and the concentration of acid affects both the number of phosphopeptides that can be identified and the enrichment selectivity. This sort of phosphopeptide enrichment chemistry using IMAP technology has been around for a while, and we've sort of confirmed what's been reported in the literature about both the type and concentration of acids and how that affects overall purification. So with AssayMAP cartridges, we found about 0.1% TFA in the sample and wash buffers gives the greatest number of identifications and best selectivity. Also looked at how chemistry can affect enrichment by looking at the amount of organic solvent, in this case acetonitrile, in the sample and wash buffers. And we've again confirmed what's been reported in the literature that anywhere between 70 to 80% acetonitrile really yields the greatest number of phosphopeptide identifications. And in addition, we've also confirmed that same composition, about 70 to 80% acetonitrile, also gives the greatest selectivity. Also characterize the capacity of AssayMAP and iron NTA cartridges. We took some yeast digest and enriched it for phosphopeptides, and we took all of the peptides that didn't bind to the cartridge, and we passed that through a new set of cartridges, and those are the golden yellow bars here. And what we can say for our model system is that we can load about 800 micrograms of yeast digest before we get appreciable signal, phosphopeptide signal in the flow through. And once we better understood the chemistry and the capacity, we applied that to doing replicates. And what we've done here is taking 500 micrograms of yeast and enriched that with the iron NTA cartridges using 4 replicates. And we identified about 2,500 phosphopeptides with an average selectivity of 96.2%. And the percent CVs are less than 5%. So very good reproducibility and excellent phosphopeptide enrichment selectivity. And if you want to learn more about how we characterize these cartridges, we have an application note that we just published that will go into great detail about the chemistry characterization that I just talked about today. And I'll close with saying that the AssayMAP Bravo and Agilent can now offer what I consider to be a very comprehensive phosphopeptide enrichment portfolio for those of you interested in doing phosphopeptide enrichment workflows. In addition to the iron NTA cartridge that we just launched, we also have a titanium dioxide cartridge for phosphopeptide enrichment. And we also have 3 different types of affinity cartridges, including protein A, protein G, and streptavidin for those of you that are interested in using motif antibodies to do phosphopeptide enrichment experiments.
Thanks, Jason. That was a great presentation to get us started, and you provided us with a nice introduction to the specifics of automated workflows and the versatility of the AssayMAP systems. Before we move on to Jake's presentation, I want to remind everyone once again to submit questions for our Q&A session at the end of the presentations. Simply type your question into the Q&A box on the left-hand side of your screen and hit submit. All of our speakers will be available to answer your questions after the final presentation. So let's get our last presentation underway. Jake, you have our attention.
Okay, thanks, Jeff, and thanks, Jason, for setting up my presentation with your excellent presentation. Today, I hope to tell you about some work that is going on in my lab that we hope will shed some light into how the internal machinery and circuitry of cells is working. Let me just give you a quick overview to today's talk. First, I'm going to give you a quick introduction to links and what perturbagen profiling looks like in the proteomics space, and then tell you a little bit more about the targeted proteomics paradigm that we're using. Then I'll get into highlighting some of the methods that Jason talked about earlier in terms of the biochemical workflows that we're using to support this very large-scale proteomic profiling project. And then at the end, I hope to give you some very early insights and tantalizing results into what we've been able to do so far. So first, let me tell you about LINKS. LINKS stands for the Library of Integrated Network-Based Cellular Signatures. The LINKS concept is to learn about cellular logic and machinery by tinkering with cells, similar to old-fashioned radio engineers who reverse-engineered radios and figured out how they worked by breaking their circuits. So in the course of my project, what we plan to do is take a number of commonly used cancer cell lines, which we'll call foundational biology cell lines, as well as some neural lineages that we're working on together with my collaborator Li Wei Tsai at MIT, and perturbing them in various ways, using drugs, by disrupting genes, or by sending cells down defined differentiation pathways that will enable us to tinker with some of these cellular circuits. From these perturbations, we'll make several types of measurements. First, we'll look at phosphosignaling through a targeted proteomic assay. We'll also look at histone marks or chromatin modifications using another targeted proteomics assay that we call global chromatin profiling. And in collaboration with another LINKS center led by Dr. Todd Golub, also here at the Broad Institute, we'll be looking at gene expression using a technology called L1000. I won't talk about that today, but hopefully you'll learn more about that in the future. Why do we need to measure all these different things? Well, we believe strongly that phosphosignaling, gene expression, and epigenetic marks are going to offer highly complementary information about how cells are working internally. Finally, we hope to be able to distill signatures from these assays in the forms of molecular profiles for each of the perturbations that we make, collapse these signatures down to representative connections between different types of perturbations, and ultimately help to decode the cell circuitry. Our center's initial questions, or initial way of tinkering with the cells, focuses on performing drug perturbations of cells. And so I've taken these three types of information that we're collecting in the signaling, transcription, and epigenetic domains and mapped them onto what we think is sort of going on inside of cells. So when we treat cells with a drug in this case, we expect there to be an early response in the form of changes in phosphosignaling, a sort of midterm response that reflects changes in the transcriptional program of the cell, and then ultimately to make these responses permanent, epigenetic changes need to take place so that the cell can adopt a new phenotype. And so we collect samples for the various profiling efforts in a time series that sort of agrees with the logic of this paradigm. We're using a few different sets of small molecules or drugs to perturb the cells in this initial round of experiments. We have one set of what we'll call epigenetic modulators, and you'll learn a little bit more about that today. A second set of compounds that are important for neurodevelopment and have been shown to be effective in management of neurological disease. And a third set of known signaling pathway modulators. Let me tell you just a little bit more about the proteomic signature assays that we're employing. Our first one is directed against phosphosignaling, and we call this assay P100. P100 is a targeted phosphoproteomic assay against 96 different phosphopeptide probes that are commonly observed and modulated in diverse cell types. And we know that these phosphopeptide probes will reflect the behavior of many different known pathways, which are sort of outlined in the circle below. And we expect that we should be able to look at just a minority of phosphocytes, because we already know that kinases are few and phosphocytes are many, and therefore we expect coordinated behavior. Our second assay is called GCP, or Global Chromatin Profiling. It's also a targeted proteomic assay, and in this case we're measuring 60 different probes that monitor combinations and permutations of different post-translational modifications on the N-terminal tails of histones, which I'm sure many of you know are extensively post-translationally modified in myriad ways. And disruptions to these modifications can create alterations in transcriptional programs and even drive disease etiologies like cancer. So I want to tell you a little bit about the targeted proteomic assay paradigm that we're using. Here, what I've shown you is a sort of typical sort of flow path for ions as they enter a mass spectrometer. And if we were to just look over time at all the ions that we were able to introduce into a mass spectrometer in a typical LC-MS type experiment, you would see a giant gamish of signal over the course of, say, the hour or two in which we do the experiment. And if we were looking for any one specific peptide or post-translationally modified peptide analyte, you could tell immediately that this sort of wave of noisy signal is not going to be very useful. But by using the mass filtering capabilities of a mass spectrometer, we can start to hone out all the peptides that don't match the characteristics that we're looking for. First, we want to select for only peptide analytes that have the starting mass to charge of analytes in which we're interested. And so, say by doing this and cutting down the ions that are allowed to enter the mass spectrometer, maybe you can start to see a peak arise from this noise that might be directed to the analyte of interest. Then we can fragment anything that matches the starting precursor mass and look for the coordinate generation of fragment ions, sometimes called transitions, that when they coalute in the course of an LC-MS experiment can guarantee proof positive identification and very sensitive quantification, because it's targeted and focused, of a given peptide analyte. And so you can see over the course of our filtering steps here, we can literally pull this needle of our peptide analyte out of a haystack. And importantly, we can do this in parallel or in multiplex and look at tens to hundreds of different peptide analytes in a single experiment at a time. Importantly, we use synthetic peptide internal standards against all of our analytes, so we get not only proof of the ID of the peptide analytes that we're quantifying, but very excellent quantification because we introduce heavy atoms onto these synthetic peptide standards. So all the quantification that we do in our assays is ratiometric against heavy peptide standards. And really, this is the technique of choice when you want to guarantee that you're looking in the right place at the right time and measure it each and every time in each and every experiment. And so the output of these sorts of targeted proteomics experiment are not simply lists of proteins that may be present in a few different samples, but fully populated matrix data where across many different conditions, and we do, say, 96 at a time in typical, we can get readouts and good quantification on nearly every single analyte that we hope to analyze in the experiment. So we propose doing these sorts of experiments to the NIH under the auspices of this LINKS program, and very luckily we were awarded this grant to perform this perturbational profiling, and we looked at our project plan and very quickly realized that this was a huge undertaking. In the 6-year course of the project, we hope to generate over 11,000 different samples, each of which will be measured in 2 or more different assays, generating over 22,000 mass spec proteomic profiles, resulting in well over 1 million individual data points. And to be able to compare experiments that we did on the first day of this project to the one that we did after 6 years of this project, we realized that reproducibility is absolutely key to analyze such a large number of samples. Let me now illustrate the workflows that we came up with in order to prosecute this project. First, I'll go through our chromatin profiling assay. Here you could see our processing pipeline takes 3 days, and I want to call your attention to anywhere where I have an orange or green dot in one of the upper left-hand corners of these flowchart squares. And you can see that even for this chromatin profiling workflow, we try to utilize automation as much as possible. In this case, we're using the Agilent Bravo LT liquid handler in combination with some 96-well extraction devices. This really enables us to get very consistent biochemical processing, again, of 96 samples at a time in parallel. And then, of course, at the end of this, we introduce our synthetic peptide standards and then perform our targeted MS experiment that I described earlier. The P100 phosphoprofiling biochemical workflow is actually somewhat more complicated, but relies more heavily on automation. So in this case, we're using a combination of Agilent Bravo LT liquid handlers, the Agilent Bravo AssayMAP device, which includes the solid phase extraction cartridges that Jason talked about earlier, as well as other 96-well devices or variable pipetting liquid handlers. And I want to just point out two important things on this slide, and that's where we introduce various types of standardization. You can see that after our initial protein digest and peptide cleanup on day 2, at that point we introduce an enrichment standard, which are a set of, again, synthetic peptides that have stable isotopes that we would call medium, in that they're maybe just a few Daltons heavier than the endogenous analyte versions of those peptides. And then after the remainder of the workflow, the phosphopeptide enrichment and peptide cleanup that's taking place on our AssayMAP automation, we then finally introduce our heavy quantitative standards, which are yet heavier than those medium enrichment standards, just so you understand the differentiation between when I say medium peptides and heavy peptides. They're just two versions of synthetic peptides that have differing isotopic labels on them. So at the very beginning, in fact even before our project began, we started efforts to automate phosphopeptide enrichment so we could do this large number of samples. And at the time that we began this, we were using, in collaboration with Agilent, custom-packed AssayMAP cartridges that had an agarose-based NTA resin. And what we would do, this resin came commercially charged with nickel on it, and we would strip this nickel off and recharge it with iron 3. And so very early on, we were having some good success. What we're able to show you here is we're evaluating the recovery of our medium phosphopeptide standards versus our heavy standards that are spiked in after for consistency and performance. And so here you can see the individual performance of two different standards across the entirety of a 96-well plate. And the general idea here is that the redder a circle is, or a well is, the more efficient a recovery was. And you can see that by and large, almost all of the wells in these plates for both of these peptides are leaning towards red, which is very positive. And then just to give a little bit of summary statistics across the entire plate, what we were able to show is that across this 96-well plate, for these two particular peptides, we had variance of around 12%, and the median percent recovery of them was around 72%. So this was great, but there were some serious challenges to overcome. First of all, we were having these cartridges custom packed by Agilent, and these were not going to be readily available either to us or to the general public, and we wanted others to be able to reproduce this workflow. Also, this agarose resin was not very robust to shipping and storage and getting it back and forth from place to place, and so that was another challenge. And furthermore, because of the agarose, the properties of the resin, they were not very optimal for working at higher flow rates. So we worked with Agilent to find an alternative. So here we're illustrating some key differences and improvements in the process when we had moved from the agarose resin, which is the sort of top line of this, to the new Agilent iMAC NTA cartridge that we were co-developing with Agilent. And you can see on top we had a scenario where we had to flow very slowly over the cartridges, and it took about 5 hours to do a phosphopeptide enrichment. On the bottom, using the new, more solid AssayMAP cartridges, we can go much faster and go through several rounds of aspirate and dispense in order to get good distribution of all of our peptides across the resin, and our total time for this enrichment is down to about an hour now. And here we can illustrate the excellent recovery across a whole plate. And we like to look at things in this plate map metric way so we get a sense of if there's any problem spots on the plate. Again, the redder a well is, the better the recovery. Here I'm now showing you an average recovery across 8 different internal phosphopeptide standards. The thing that looks like a little H in the middle of each circle is the width of that H is proportional to the variance. It's kind of like an error bar there. And then you can see we had one frank failure in this plate in well E1. And so that's great. Looking at this plate in this way, we can immediately discard that sample, and we can feel very comfortable with the performance of the phosphopeptide enrichment across the entire rest of the plate. And just some more general summary statistics that I can give you here. These are just representatives of the 8 different internal standards that we're using, and our CVs range anywhere from as low as 12% to up to 22% in some cases, but generally in a quite acceptable range for our, what I would call, research-grade assays that we're performing here, as opposed to a clinical assay, which might require higher performance. And just one further look at the quality that we can obtain here. For very relatively low input phosphopeptide enrichments, and we put 500 micrograms of protein lysate digest into our workflow. You can see just now by standard DDA or shotgun proteomics, we're pretty much able to get somewhere around 5,000 distinct phosphopeptides in any given well, and the specificity in terms of percentage of recovering phosphopeptides as opposed to peptides in general, non-phosphopeptides, is quite high, around 94 or higher percent from an intensity weighted average of these things. Okay, so now that I've described our process to you, I just want to give you a very quick look into the data that we've been able to get so far on this first set of perturbations, which are epigenetically active compounds. We performed these perturbations in a couple of different cell models, a few cancer cell lines representing different types of cancer, and a neural lineage, which is neuronal precursors that we had derived from human ES cells. So all in all, we had 5 lineages treated with 32 different compounds in biological triplicate across 2 different assays. It's around 1,000 samples, each generating its own molecular profile, and this is over 70,000 individual data points. You can look at and explore all of these data in our web-based targeted mass spec repository called Panorama. The link is there below. This software is developed by Mike McCoss at University of Washington, another collaborator on our project. He has also designed the Skyline software that we use to do the initial quantification of all of these data. And Panorama is a really great way to host our data in the web. And so I'm going to talk a little bit about how these epigenetically active compounds are affecting phosphosignaling states now. So at the end of the day, what we do is assemble these things called connectivity maps using our data. And these are based on the correlations between different perturbations in given cell lines. So let me just take a moment and try to orient you to what's going on here. In each case, one circle in this diagram is a particular perturbation in a single cell type. And the inner part of the circle is colored by the compound class to which the compound belongs, and that's illustrated with a legend on the left. And then the outer part of the circle, when there's one there, is colored according to the cell line in which the perturbation was done. You'll see there are some gray circles with black lines around those. Those are just simply things that we wanted to blank from the annotation for purposes of clarity here. Now, what you can see is that unexpectedly, is that these compounds shaded in green that are sort of in the center of your screen here, these are all histone deacetylase inhibitors. And we certainly expect and do observe strong signatures of these things in the chromatin profiling assay. But here we're looking at a connectivity map that reflects phosphosignaling changes in these different cells. And so any time that I've drawn an edge between any two of these nodes, that means that there is a pretty strong correlation of at least 0.5 or better between those two treatments. So it was somewhat surprising to us that we could observe these connections of HDAC inhibitors in a phosphosignaling assay, and that there are other sorts of inhibitors also exhibiting strong connectivity, including compounds that affect the enzyme G9A, which is an enzyme responsible for putting methyl groups onto lysine 9 of histone H3. And furthermore, that in space, these perturbations that affect G9A not only cluster together, they also cluster sort of together with these HDAC inhibitors. So what we're seeing is sort of in the phosphosignaling space an interplay between epigenetic processes or epigenetic enzymes that are responsible for the deposition of methyl groups and acetyl groups onto histones. We're not quite sure what this means yet, but we're pretty excited about this novel result. Then I've just got more data slide for you, and this is a big one. I'm going to try to explain this one as best I can. So here I'm showing you pretty much all of the data that we've generated so far. Just like on the last slide, each node is a particular condition where the inside is colored by the compound class, the outside is colored by the cell type. And now what I've done is color-coded all of the edges between the nodes. So if an edge is blue, it means that this particular edge arose from the global chromatin profiling assay, and if an edge is pink, it arose from our phosphosignaling P100 assay. And what I hope you can appreciate from this very tangled diagram is that a lot of connections that run between cell types, and you can see here that I've organized all the nodes sort of in circles of their cell types. All the connections, or many of the connections that run between cell types arise from the global chromatin profiling assay, whereas many of the connections that arise within a given cell type, and maybe just focus on the MCF7 in the lower right for now, seem to be arising from our phosphosignaling assay. It's not an all or none sort of thing, but it is sort of intriguing to speculate that the internal wiring of cells, which is governed in this case by phosphosignaling, is very cell type specific, but yet all roads sort of lead to Rome when it comes to the final state of the chromatin, and we can get effects that are very similar in the chromatin assay that seem to propagate across cell types. And in fact, we've quantified this a little bit, and we can sum up all the edge weight that either happens within a given cell type or goes between two cell types, and we find that about 60% of the edge weight within cell type arises from P100 data, whereas about 60% of the edge weight that goes between cell types arise from the chromatin data. So in summary, I hope I've showed you that the proteomics-based molecular profiling is a viable experimental strategy, and it's an important tool for decoding the internal wiring of cells. Now, in order to do such a large project, of course, scalability and longitudinal reproducibility was very important, and we benefited immensely from having automated workflows. Development of these automated solutions for phosphopeptide enrichment and for the chromatin profiling assay as well were actually simply a prerequisite for being able to do studies of these scales. And that our early insights from this phosphoprofiling and chromatin profiling efforts at this scale reveal some pretty intriguing possibilities about how cells are internally wired. I'd just like to thank my group and the folks at Agilent who helped out with this tremendously, as well as NIH for the funding, and thanks to Jeff and GEN for the opportunity to share this work with you.
That was excellent. Thanks very much, Jake. I think our audience now has a great sense of the importance of proteomic-based molecular profiling and how automated systems can streamline workflows for a variety of laboratory scenarios. Before we get started with the Q&A session, I want to let everyone know this is your final chance to submit your questions for speakers. It looks like we've already gotten some really great questions, so keep them coming, and I'm encouraging the audience to keep even more of them coming in. All right. Since we have a few questions already, and they look really good, let's begin the Q&A session, and we'll try to get to as many questions as we can. Okay, our first question is for Jason. Jason, one of our members would like to know, you offer multiple cartridge types for phosphopeptide enrichment. Which one is the best?
That's a good question. The reason that we offer several different options is because we don't know which technique is the best, and it really is probably going to be dependent on the model system or the system being studied and the desired analytical goals. I know in terms of global phosphopeptide enrichment, in my hands and the model systems I've used, I've had the best success using IMAP chemistry, iron NTA. Certainly, if you browse the literature, you'll see people having great success with titanium dioxide and using IMAP chemistry with other types of transition metals, including gallium and zirconium and titanium. So those, I think both of those work really well. My best suggestion to customers is to just try them out. Now, if you're looking for more targeted types of phosphopeptide enrichment, then using the motif antibodies is also a really good choice. That gives you, I know there are companies that manufacture anti-phosphopeptide antibodies so you can target certain pathways. And so my suggestion, again, would just be if you have the opportunity, try them all. But in my hands, if I had a first option, I would try iron NTA.
All right, thank you very much. Our next question is going to be for Jake. Jake, one of our audience members would like to know, they asked, does your assay and automated workflow see mostly serine and threonine phosphorylation? If so, why did you choose to focus on these?
Oh, yeah, that's a great question, and indeed, as I'm sure many listeners and watchers are familiar with IMAC technology, the fact that it does tend to enrich primarily serine, threonine phosphosites on peptides. So in fact, the assay does tend to focus on those classes of phosphopeptides. And the reason we chose to focus here is for really, really there are a variety of reasons. So one is there are several very good and well worked out assays that focus specifically on tyrosine phosphorylation. A lot of effort put into studying tyrosine phosphorylation, the tyrosine signaling pathways. But yet the majority of phosphosites, phosphoproteome, belong to threonine phosphorylation. So we felt it was sort of an underserved area, and there are obviously smatterings of assays that are dedicated to certain phosphosites, but we wanted to develop something that was general and useful for this really large class of underserved events.
All right, thanks very much, Jake. Jason, another question for you. What are the most important chemistry considerations for successful phosphopeptide enrichment by IMAC?
Well, what we found and what other investigators have found is it's really fine-tuning the control of pH. And the pH isn't as simple as titrating in the amount of acid. What I found is that what really controls the degree of ionization of things that are acidic is the amount of organic solvent. So really the key to successful IMAC enrichment is maximizing the degree of ionization differences between phosphoryl groups of phosphopeptides and carboxylic acids. Because if carboxylic acids are deprotonated and are negatively charged, they'll also bind. And it's very tricky just to set the pH using acid alone, and that's really the purpose of the organic solvent. I think a lot of people think that 70 or 80% acetonitrile are in the solutions solely to reduce nonspecific binding by hydrophobic interactions, but really that is what fine-tunes the pH. So I think the most critical key to getting the largest number of phosphopeptides and getting good selectivity is to really pay attention to the pH, which is very strongly correlated to the amount of organic solvent that's in the buffers and in the sample.
All right, thanks very much, Jason. Jake, one of our audience members asks, so do you think that phosphorylation event is a prerequisite to epigenetic perturbation?
Well, that's a really interesting question. I think that it's way more complicated than we think. I don't think that phosphorylation events are necessarily a prerequisite to epigenetic perturbation or changes in the epigenetic states of cells, but I think that there are maybe many concomitant signaling changes that occur in cells at the phosphosignaling level when the epigenetic machinery is perturbed. And then, sort of interestingly, in a study that we did in the lab, I haven't showed data from it here, we did find that there are sort of blocks of proteins that are known to regulate the epigenetic machinery whose phosphorylation state changes in response to a wide variety of drugs in a highly coordinated manner. So I think, one of the main questions of our project is, what is the interplay between two different signaling systems in the cell, the sort of phosphosignal and epigenetics? Hopefully, throughout the course of the project, we'll be able to shed some light on it.
All right, thank you very much, Jake. Jason, another question for you. It asks, I've read materials from Agilent saying that AssayMAP Bravo can process 8 to 96 samples. Can I process fewer than 8 samples using AssayMAP?
Yes, you certainly can. Most of our literature, we state that the minimum number of samples is 8, and that's primarily because historically the people interested in automation are high throughput types of customers. And they visualize the deck or the sample plates in column and row fashion. And when the AssayMAP head was designed, we designed the head so that the amount of pressure that's applied to properly seat AssayMAP cartridges is applied in increments of 8 or row increments. So the prerequisite is that you need to have multiples of 8 cartridges on the deck to be properly seated, but those cartridges don't have to be functional cartridges. We offer cartridges that are, we call resin-free, that don't have any stationary phase packed in them, and we use those as placeholders. So in actuality, if you're not in the high throughput camp like Jake, and you only have several samples you need to process at any given time, you can design your experiment using all 96-well plates and only have one real sample and use, for instance, an iron NTA cartridge at one position and have 7 resin-free cartridges at the other positions and use the automation to process one sample. But in terms of layout and the way that we write our protocols, it definitely is in a column-based format.
All right, thank you very much, Jason. Jake, another question for you. It asks, how can you account/correct for the variability in phosphopeptide recovery across the 96 sample system?
Oh, that's another really excellent question. So there's a couple of ways that we deal with this. First of all, through the use of our enrichment standards, which are spiked in prior to the phosphopeptide enrichment comparison, with the quantification standards that are spiked in after the phosphopeptide enrichment, we have measurements for this quantity across every well of a 96 well plate, and so we can use the recovery information in order to perform corrections on the data that is linked to the actual recovery for that particular sample. And we do some other sort of loading correction metrics as well, using the characteristics of the whole data, but knowing the recovery in each well is certainly key and correlates very highly with the loading corrections that we do. I'll just add that, of course, in moving to any higher throughput technology, and especially in proteomics, tends to be finicky, as many practitioners test. It is important to build in the proper process monitoring steps in order to make these sorts of explicit corrections and find out where variability are. So thanks for bringing up that important.
All right, thanks, Jake, but don't go too far because we got another question for you. This next question asks, what is the phase and reason for an SPE in plate and not AssayMAP cartridges?
That is simply a volume processing sort of consideration. In the first stages of our automated processing, while it is performed on an Agilent Bravo LT liquid handler, our digestions are performed in scales that are more like a milliliter of reaction mixture, and that sort of volume is not the best match for the AssayMAP cartridges. So in this case, in order to do our cleanup on just after the proteolytic digestion, we move to a 96 well SP plate. That's Waters Oasis or that packed plate, something like that. So it's a reversed phase like resin that we do again in parallel using positive pressure extraction manifold. Now, once we've completed that, of course, we dry down all the peptides and we can bring it up in volume that are then much more amenable to processing AssayMAP cartridges.
All right, and let's take about maybe one or two more for you, Jake. Some more questions about your organization and team. It says, where can I find out more information about links and how can we become involved?
Oh, great. So links, the overall clearinghouse for learning more about links is at linksproject.org. Again, that's linksproject.org. As I mentioned during the presentation, LINKS is an NIH-funded program, and if you go to linksproject.org, you'll learn about the 6 data generation centers that are part of the LINKS consortium, as well as the data generation or the data coordination center that's helping us stitch all these data together. And so, in addition to our Center for Proteomic and Epigenetic and Signaling Profiling, there's a Center for Cryptomic Profiling. There's a center that's quite concerned with how microenvironments affect the growth of cells. There's a center that's interested in cardiotoxicity of common therapeutic agents. There's a center that is very interested in ALS, or more commonly known as Lou Gehrig's disease, and they're applying perturbations to motor neurons that they derive from ALS patients or healthy controls. And then there's another center that's quite interested in the sort of flip side of my coin, looking at how tyrosine signaling pathways are activated in cells through a variety of standard lab assays and imaging assays. And so if you go to linksproject.org, you can learn more about each of these projects, as well as find out how to access data from part of them. All
right, and I think this is going to be our last question. Again, for Jake, it says, where can I learn about the specifics of your assays, and can we do them in our labs?
So our chromatin profiling assay has been well described, been referenced in a number of publications. We have a really excellent publication in Methods. The first author is Amanda Creech from my group. So if you look for Creech et al. Methods 2015, it really describes all the nuts and bolts of the chromatin profiling assay and what you would need to do it in your own lab. I hope to be able to say that the same thing happens for the P100 phosphosignaling assay, and that a publication is following close behind. There's just that matter of the pesky reviewers who need to accept our paper journal, but hopefully that will be coming not too long from now. But the other thing I wanted to say is that, especially for this P100 phosphosignaling assay, there's very little barrier to people executing the assay in their own labs. In fact, we acquire the data using a pretty simplistic data-independent approach or data-independent acquisition approach for the mass spec part of things. And we can envision either shipping collaborators pre-made mixtures of our peptides, or even maybe having some third-party company sell a mixture of all the peptides that you need to execute the P100 assay in your own lab. And as long as you can acquire data in a DIA or swath-like manner on your mass spectrometer, we could then follow on with providing templates for quantification of those data in Skyline. And you could not only run the assay in your lab and compare it to all the assay data that we're collecting as part of the LINKS project, but you could sort of turn that around and contribute to the LINKS project yourself if you're interested in running the assay in your lab. And I would be happy if folks would contact me about the possibility.
Great. Thank you very much, Jake. That was an excellent answer there. So with that, everyone, we've come to the end of our webinar. And I'd like to remind everyone that the webinar will be archived for 6 months on our website at www.genengnews.com. So if you missed parts of it, or if you want to watch it again from the beginning, feel free. Or you can forward the link to your friends and colleagues, which we highly recommend. I'd like to thank Jason and Jake again for their informative presentations, and I'd like to thank the audience for their attention and thoughtful questions. And a very special thanks to Agilent for sponsoring this webinar. So hopefully we'll see you again soon at another Gen webinar in the near future. So goodbye for now.